Hi, I'm so happy to have you here, Dr. Barbara Bailus. I am Dr. Terry Jo Bichell, and I am a neuroscientist, as you know, and the mother of a son with Angelman syndrome, as you know. And I am very, very excited to be able to ask you what you've been doing with FAST for the last couple of years, because I miss you and I haven't talked to you in a while. So if I could get you to introduce yourself and just tell us a little bit about you, and then I'm going to start asking questions.
That sounds great, Terry. Let's see. I guess for the audience that doesn't know me, so I did my graduate work on Angelman syndrome, and I worked with the artificial transcription factors and turning off the antisense in the mouse, thus enabling the regular UBE3A on the paternal side to turn on. And I did this in the laboratory of Dr. Dave Siegel. From there, I went on and did postdoc work actually on Huntington's disease, but still staying in gene editing. And I'm currently now an assistant professor at the KECK Graduate Institute, one of the Claremont colleges in California. My work with FAST began back when I was a postdoc, where I first started helping do some scientific advising, and then moved forward to where I did some scientific communications. And now I currently serve as the chair of the Scientific Advisory Board for FAST, which has been a great honor and which I really enjoy.
That's so cool. I saw that announcement that you were named as chair, and I just really excited to congratulate you. And tell me, what does that job entail?
Yeah, so no, we don't actually get a special chair, even though it sounds like we should. But what it does entail is that I actually do a lot of sort of looking at the grants that come in for FAST to fund and finding the correct reviewers on our scientific advisory board so that they can then get a review, and then the board of directors looks through all the reviews that the scientific advisory board has put together and decides on whether to fund or not fund specific grants. In some cases, I will also serve as a reviewer, but in many cases, it is just looking and finding the correct expertise for the review.
Oh, that's great. So you are in the enviable position of knowing about all the projects that are coming in and applying for funding, which must be really, really fun.
It's very exciting, especially with the cutting edge technology that FAST is looking at sponsoring and funding, to see it sort of in its phase, and then to realize like, okay, this could progress all the way to an actual therapy or treatment. So it is a really wonderful position to be in.
So how do they uh, decide which, besides the fact that you're having the applications reviewed for scientific rigor and the credentials of the person sending in the application, all those things, how do they prioritize what to fund? Who decides what kind of applications are even going to be looked at?
Mhm. So, I mean, FAST's main priority has always been curing Angelman syndrome. So there's a heavy emphasis on translatability for the applications, so that we are pushing forward to a therapy. And we often look at that. That's one of the criteria that the reviewers are asked to review on, is what is the translatability of this moving forward? And as mentioned, we have about 3 reviewers for every single grant. And then with that, when it goes to the board, you also have Dr. Brandt, who is looking at it as well. And then there's discussions about, funding, what level to fund at, and so forth. So it's a very collaborative process to make sure that everyone gets a fair review.
So I just want to define translatability, because that is a little bit confusing. You're not talking about translating from English to German or Japanese to French. What are you talking
Mhm.
about when you say translatability?
Yeah, so this is a term that's bantered about and quite hot in the field, which is translational science. And what that means, you've probably also heard the term from bench to bedside. So when you think of it, you know, we as scientists do a lot of work on what we call the bench, which is with our test tubes and our pipettes, and we're moving clear liquid from one tube to another. At some point, hopefully that moves into cells and then into mice and then into humans. And so there are a lot of ways in which we start to think, how could we take what we're doing on the bench and move it along that pathway so that it eventually translates from the bench and your test tubes into a mouse, and then, because mice are not humans, into a human, or in this case, an Angelman syndrome patient. And so that's one way in which we look at this, is what impact will this have on the patients as we move forward? So there's a
Cool.
lot of interesting science that can be done, but not all of it maybe necessarily is as apparent to be translatable at first glance.
So that sort of differentiates basic science from science that is going to maybe actually treat a living patient. That would be the difference between basic science and translational science, right? So when can you
Yeah, I mean,
can you think of
oh,
a
sorry, no,
project? Yeah,
finish,
sorry,
please.
there must be a lag, so I'm talking over you. Sorry about that. Can you think of an example uh, of a basic science finding in Angelman syndrome that uh, would be interesting to a scientist, but might not be translatable?
So I think probably some of that work, when you look at some of the early work that's been done, you see some of that, you know, with basic. There are some cases where we think more, you know, this could translate at some point, but it's not that far along, or this is really interesting. So maybe like a crystallization structure, or a, you know, analysis of how precisely every single isoform of an RNA would fold up. Those are maybe what I would consider basic to start with. Now, of course, understanding that can lead to translational therapies, but it's a longer journey to get there. So it's something that has potential, but maybe isn't something that we're looking at being a therapy in the next, you know, 5 years.
So in an example of a translational um, proposal or a translational project, can you think of one, maybe recent one that already got funded that you could talk about?
Yeah, so I think a really, you know, a couple of recent ones that you can see. So there's the one that is already in current clinical trials, which was work that Scott Dindo did several years ago, where he was looking at using different ASOs to, you know, block the transcription of the antisense for UBE3A that results in the paternal activation, and thus that is very translational. You can see that as being something that can be applied, and its main intent is to be a therapy. And so that now, of course, is in the phase 1-2 trials. There are others where I would say like a small molecule screening, where you're looking for UBE3A activation in cells, is very translatable. Jo Anderson's work that was done at UC Davis in collaboration with the Siegel Lab and the Silverman Lab. And what they're doing there is they're able to modify hemopoietic stem cells, which are the stem cells that make up your bone marrow. They have it in such a way that these cells are going to express UBE3A, and the cells are also able to get through the blood-brain barrier into the brain, where this UVE3A is needed. They've done this in mice, so this is not in humans yet, but what you see is a very clear translational ability where you're going from something that was in theory a possible treatment into a mouse and then showing amazing results in behavioral gains. And so that's also another good example of a translational project.
Nice. That
I
was a
think I got
great
that right.
example. Yeah. And also we interviewed him recently, so that'll be good to have both your explanation and his explanation. So a couple questions. One is, do you ever get a an application out of the blue that just has a crazy idea that you never thought of or nobody you know has thought of that is like a great idea?
So I would say that we get a lot of applications, and there are some where I look at and I'm like, this is going to really push things forward or be amazingly helpful. But I wouldn't, I don't know if terminology for it. So, but I have seen applications that are, to me, are very, very exciting. Joe's probably was one that was very exciting when it first came across, to see sort of that process. So.
And it was something that nobody else was trying. Um, so how do you fit this in with your regular job? Because you're also the assistant professor, and what do you do in that capacity?
So in that capacity, as an assistant professor, I teach my classes with my students. And then I also have service to the university that I do. And then I also, of course, am currently in the process of starting up my own lab, which is really exciting because that took a little while due to COVID. So I'm now in the process of that. And so that is a good portion of my week. With FAST, so our university has where they call service to the profession time, to the field time. And so we're allowed a certain amount of time in which we can actually dedicate to doing that to the profession. And so that is where I fit in the time for FAST as well.
Wow, that's great. So it's actually part of your job, the service to the profession. So what are you going to study in your lab?
So I probably will stay with neurological disease, since that was what I spent both of my doctoral and postdoctoral work, and of course Angelman syndrome being one of those. Also gene editing, and probably a little bit on delivery mechanisms as well. So that was something that I studied in both labs, was how to develop and design gene editing proteins that can get into the neuronal system.
Hmm, cool. So, I mean, drug delivery, it is trickier than people think in terms of how do you get a compound or a gene or an antisense oligo or whatever it is you're trying to get through the blood-brain barrier to the neurons where we need it to go. And so that's pretty complicated. So that's what you're going to try to study. In
So I'll
my
be doing, so to start with, of course, I'll start with cells. And then eventually I will probably move into mice, because if you want to study a blood-brain barrier, at some point it really helps to move into an organism that has a blood-brain barrier. And mice are definitely one of those very first ones that you look at. So yes, one day maybe mice.
So how did you get interested in this whole topic in the first place? I mean, before you did your PhD, what made you even study this at all? And you were doing Angelman and gene editing through your entire PhD, right?
Yes, so I was doing Angelman and gene editing through my entire PhD. So if we go back before my PhD, which you were asking about, I would say that I entered college with the idea that I was going to be an environmental lawyer. Uh, and then spent some time in one of the classes reading a bunch of case briefs and deciding that that was probably not the career track that I wanted. So I asked myself, what classes did I really enjoy? And I really enjoyed my laboratory classes. They were one of the only things that I would get up early in the morning for, because I'm not a morning person. And so I switched my major at UC Davis to biotechnology because it had the highest amount of lab classes that was required to be taken. I went into industry for about 3.5 years at a small startup company. It no longer exists, but it was called Cytograph Tissue Engineering, and did a lot of tissue culture. And they actually were going through phase 1 human clinical trials where we would implant lab grown tissue vessels into hemodialysis patients. It was very exciting. But at the time, I learned a couple of things about what I wanted for my future career, which was I love training our new employees, so I like teaching. And I also wanted to design my own experiments. I talked to my mentor there at the time, and she said, well, if you want to do both of those things, you really need to go get a PhD. So then I started looking at PhD programs, and my love of biology really had centered on genetics. And so I started looking at genetics programs. And when I interviewed at UC Davis, I actually met Dr. Dave Siegel and was talking to him about zinc finger technology and gene editing technology. This was way before CRISPR had come out. And I decided that I would want to join his lab. And so that's how I arrived at UC Davis. And when I arrived there, he had just received a small grant to do research on Angelman syndrome. And I was the new graduate student in the lab that took on that grant. And we were very fortunate. It progressed from there into being a study that we got NIH funding for and then FAST funding for. And it went all the way through and got published in 2016.
Wow, that's so cool. So it was serendipity meeting David Siegel and then moving from there. So one thing they used to have the fire team at FAST with the 5 scientists always sort of talking to each other and bouncing ideas off. And so give me an example of how some of the funded scientists are able to collaborate.
Yeah, so that's a great question, Terry Jo, because FAST is an extremely collaborative environment. And so what we have is we have several projects that are going to benefit across multiple laboratories. So we have over at Yale, Dr. Zhang's lab, where he's actually getting different genotypes of stem cells from different Angelman patients, and he's making a biorepository of those stem cells so that other researchers in the Angelman field will have access to them. And for example, Dr. Keong is actually taking some of those stem cells and making organoids from them, which are like mini tiny brains. And so what you can do there is then use it to test different small molecules or other potential treatments, or even study just some basic differences between the genotypes. And then we also have over at UC Davis, Dr. Siegel's lab, where they have an infrastructure for doing testing in Angelman syndrome mice and rats, and that can benefit researchers who may not either have that expertise or the facilities, but have a really promising treatment that they'd like tested. So you can see this huge collaborative network that FAST has enabled to be established for Angelman syndrome.
That's really cool. Really, really, really neat. So there's another thing I know about you and that is that you really like to be able to explain science to the general public. And I think you're really good at that. And not only are you good at it, but it seems like you really like trying to do it and to make science understandable. And can you just tell me, why do you like doing that and how do you do it?
So I think why one of the basic things that I've always believed is that NIH is hugely funded by tax dollars. That is what it's funded by. And so that means that the public is funding our science. And I feel that the people that fund our science should have the ability to understand what it is that we're doing. And I also understand that sometimes it can be really challenging, especially with the amount of scientific jargon that we will use to describe our science. So I always enjoy being able to break it down to allow people that are actually funding our science to better understand what it is that we're doing. I think this partially comes from the fact that neither of my parents are scientists. So I did indeed spend most of my PhD and postdoc career breaking down the concepts so that they would understand what their daughter was doing all the time. And I did a lot of work in my postdoc career with outreach and public, science talk so that they could understand, what it was that the Buck Institute was doing and what my research was as well. And currently at KECK, I teach a course in scientific communication where I help the students understand what different scientific audiences you might have and what their background might be coming into it. And I think that really helps. It gives the students sort of a perspective and it makes them more comfortable with understanding their scientists. And I think it also establishes a certain amount of respect because I always remind them, just because they don't understand the scientific talk doesn't mean that your audience isn't a bunch of intelligent, highly sophisticated professionals. They just have that in a different field. So I asked them, imagine if you were going to something where they were discussing, law, you're not going to understand a lot of what's being discussed there, but you have a PhD. So we have to get everyone to be able to understand each other and be on the same page. And I think that also helps just in general for the public, realizing how science is helping, how it can advance different things and treatments.
You're amazing and you're doing so many things, and you probably don't have any free time to have fun. And we have been having this pandemic lately, but what would you do if you had some free time?
True. So free time is at a premium, but let's see, things that I do when I'm not at the bench, not grading papers, and not looking over grant possibilities for FAST. So I love to do art. So I paint in my spare time. I also bake and I garden. And when I'm feeling more athletic, I enjoy horseback riding. So those are the areas. And when I have a free weekend, you can usually find me at the beach.
Wow. It's a good thing you live in California. You can do all those things in California. That's fantastic. So, well, thank you very, very much, Barbara. It was so nice to talk to you and see you even virtually, just for a minute. And I'm just excited about all the different things you're doing. And I really appreciate you taking the time. So thank you.
Thanks, Terry Jo.
Bye.